I'm Sarah Mizielinska and I'm a group leader at the UK Dementia Research Centre at King's. My research focuses on how a protein called TDP43, which is found aggregated and mislocalized in the brains of people with motor neuron disease and dementia, how it gets there, why it becomes mislocalized, and really kind of focusing down to the molecular level of how things go wrong. In motor neuron disease, we know that nerve cells, both in the brain and also in the spinal cord, become dysfunctional. And one of the main things is that transport through them can't keep up with the demand that those neurons need to communicate with each other. And that's how it leads to the clinical symptoms such as kind of not being able to use your muscles properly. And ultimately, that process leads to further and further neurodegeneration that we see in disease. In order to study how the movement of things becomes dysfunctional in disease, we really require high-level imaging facilities. And here at King's, we're really fortunate to have a world-leading facility for microscopy. And that allows us to study human brain tissue and then all the way down to studying single molecules moving within cells. And within my research group, we're using this to study how things move across the barrier between this nucleus, where the DNA is, and then the rest of the neuron, and looking to see how single molecules move across this barrier and really the very nitty-gritty of how that becomes dysfunctional in disease, and then to understand how we can try and improve that to stop disease spiralling out of control. Being part of the DRI has been enormously advantageous. So what it has allowed me to do specifically is really to do high-risk, high-reward work, which I wouldn't have been able to do otherwise, and really to answer some of the critical questions in disease, such as kind of this mislocalization of this protein TDP43, which is the major cause of most motor neuron disease and a large part of dementia as well.